Hello, my name is Madison Kane, and this summer I worked in Professor Linda Griffith's lab. My research supervisor was Raphael Zonis, and the research project I worked on was designing an optimized protocol for loading of cryoprotective agents and the vitrification of human oocytes. So vitrification is a procedure to store oocytes for long-term storage purposes. It's also an assisted reproduction technique that women may choose to undergo in order to preserve their fertility due to an upcoming cancer treatment, gynecological surgery, or genetic issues. Or women may choose to undergo this procedure in order to postpone their pregnancy due to career or financial reasons. Different companies make vitrification solutions and systems. However, the overall protocol for vitrifying oocytes is the same. Women who have undergone ovarian stimulation will have their oocytes harvested, and then the oocytes will be loaded with cryoprotectants. In order to load the oocytes with cryoprotectants, the cells are placed in a vitrification solution that is composed of a carrier solution and different kinds of cryoprotectants. The carrier solution is a delivery mechanism to deliver the cryoprotectants to the oocyte, but it is also composed of different components that are also essential to the oocyte. And the different types of cryoprotectants that are used are non-permeating cryoprotectants, which do not enter the oocyte, but are essential for the vitrification process. And then there are also permeating cryoprotectants which are used, which can penetrate the oocyte and help prevent intracellular ice formation. Intracellular ice formation can occur during vitrification and can cause damage to the cell membrane and also to different types of organelles within the cell. So it's important to minimize this intracellular ice formation by loading the oocyte with the permeating cryoprotectants. So once the oocyte has been successfully loaded with cryoprotectants, the cell can then undergo vitrification in liquid nitrogen for storage. And then when women are ready to retrieve their oocytes, the cells can be warmed and the cryoprotectants can be removed. And then the cell can undergo in vitro fertilization. Potential challenges associated with cryoprotectant loading include osmotic intolerance. So when cells are placed in different concentrations of cryoprotectant, they can undergo cell volume changes, and this can potentially be damaging to the cell. And another challenge associated with cryoprotectant loading is cryoprotectant toxicity. So when oocytes are exposed to high concentrations of cryoprotectant, this can sometimes be damaging to the cell. And if the concentration is too high, it can even in some cases cause cell death. So in order to minimize the chances of these situations occurring, it's important to develop an optimized protocol that takes into consideration the concentration of the cryoprotectants used, the time of exposure, and the temperature. So this summer, my goal was to conduct a literature review on the properties of cryoprotectants, the different types of commercial vitrification solutions used, and different cryoprotectant loading protocols. And we hypothesized that data was available on loading cryoprotectants, however, the data was scattered. So by performing a literature review, we could help to synthesize the information and gain an insight on how to design an optimal loading protocol. So from our preliminary findings, we found that a mixture of permeating cryoprotectants is beneficial and can help reduce toxicity to the cell. We also found that non-permeating cryoprotectants are important for the vitrification process. And we also found that equilibrating oocytes with lower concentrations of cryoprotectant prior to exposing them to the higher concentrations within the final vitrification solution can help the cells during the procedure, and this is an important step. And different types of mathematical models can be used to find optimal times of exposure. And also the composition of the carrier solution is important for the vitrification process, and finding the correct salt concentration can really affect the cryoprotectant loading. So we are currently utilizing the information we found in the literature review to design a loading protocol, where we take into consideration the vitrification solution composition, time, temperature, and exposure profiles. And we hope that by optimizing the protocol, we can increase cell survival and viability post-cryopreservation. So this is the end of my presentation. I would like to acknowledge the MSRPx program, Professor Linda Griffith, Raphael Zonis, Dr. Joseph Doyle, Dr. Nicole Faithful, Cryotech, and the Griffith Lab. And here are my references.